Hey, Bio30s, and welcome to my first of two discussions about the evolution of genetics as a science in the 20th century. Once Mendel passed away and did his experiments in the 19th century, his work sort of got kind of lost for a while there. There weren't a lot of people that were really paying attention to what he had said. And it wasn't until the early 20th century when scientists began to kind of rediscover the significance of what he had done in the 1800s. And if you want to take a quick peek at that, just to kind of refresh your memory and to see some of the players that were involved in the early 20th century, give that video that discusses the history of I guess, Mendel's contribution to genetics and how it sort of began to flourish in the early 20th century. That would be a good video to watch there. But one of the guys that sort of came out in the forefront in the early 20th century was a guy by the name of Thomas Hunt Morgan. I've referred to him already. I briefly discussed him when we discussed the history of the history of, I guess, looking at genetic material and the history of trying to figure out what DNA was and what its structure was. I sort of referred to him very briefly, and I said that I would get back to him later in the course. Well, I'm getting back to him now. He did some very important experiments early in the 20th century that began to kind of shed new understanding on some concepts that Mendel had never even considered. I mean, at this point now, there was a much better understanding of nucleic acids. At this stage, we're not sure that it was, the scientists were still not sure whether or not it was genetic, it was DNA or proteins that were transferring genetic material, but there was a much better understanding how the transmission occurred, and a lot of that had to do with Morgan's work. Now, Morgan started something that has been now a keystone of genetic research ever since. He began using a species known as Drosophila melanogaster, which is basically a species of fruit fly. And geneticists still use fruit flies today. They're very common in a lot of genetic labs around the world. Now, why is this? Why are fruit flies such a great species to study when you're dealing with genetics? Well, a couple of things. They have a rapid reproductive rate. They can lay more than 100 eggs per mating. They can reproduce very shortly after hatching. They have a very short life cycle, only 10 to 15 days in length. That allows many generations to be studied in a very short amount of time. They have very small size. They're very small organisms, so you can be housed in very small conditions, like a test tube with a very small amount of food. You can have lots of these flies that are being basically reproduced, and you don't need a large area to kind of house all these organisms while you're doing your experiments. And there is a sexual dichotomy, which makes it very easy to distinguish the males from the females. The males are very easy to identify from the females, so it's kind of easy to kind of figure out, who's who when they're dealing with, who's mom and dad in these experiments. So that was sort of the reason why he began using fruit flies. And when he began using his fruit flies, he began doing experiments like Mendel would have done with his pea plants. And he, at one point, he discovered that alleles of two different genes on the same chromosome don't always behave the way they should. He understood that there were what were called linked genes, that some genes on a single chromosome would be inherited together as a unit. So if you got, in the case of him, he basically did an experiment with the genes that controlled eye color for fruit flies and their wing length. So he began crossbreeding them. So he had recessive, he had fruit flies that were, they were homozygous, pardon me. They were homozygous for both recessive traits. So they had purple eyes and they had vestigial wings, or kind of really short wings. And then he bred them with heterozygous dominant flies that had normal colored eyes, but were heterozygous for the trait. And they had normal wing length, but were heterozygous for the trait. Now, as you can see on the slide here, if you run that Punnett square using this set of alleles and this set of alleles, if you run that in a Punnett square, in theory, what he expected to find when he bred these parents is that he would have had a 1-to-1 to 1-to-1 ratio. He would have had a quarter of his offspring would have been purple vestigial or purpleized and vestigial wings. He would have had 1/4 of his offspring that would have had normalized and normal wings. He would have found that a quarter of his offspring would have had purpleized and normal wings, and he would have expected to find that a quarter of his offspring would have normalized but vestigial wings. But he didn't get that. What he got instead was a ratio that was way off. He got a ratio that was basically a 9-to-1 to 9-to-1 ratio. What he found was, and I've got him indicated here with asterisks and little triangles, he found that 90% of the offspring that came out from this pairing looked like one of the two parents. So he found that about 45% of the offspring were purple vestigials, and then he found that about 45% of the offspring were normalized with normal wings. The remaining 10% was divided, 5% each, with the offspring that had combinations of traits from both of the parents. So 5% had purpleized and vestigial wings, and the other 5% had normalized with vestigial wings. So that was sort of what he discovered. Now, he didn't really know how to kind of initially, I guess, justify this, but we already know the reason why this occurs. This is something that we have talked about already in this course. When gametes are formed, thinking back to meiosis 1, you remember we talked about how genetic recombination occurs in the gamete. We talked about independent assortment, where the chromosome pairs will flip here or there. You don't know, it's a flip of a coin for every pair of chromosomes. Which chromosome is going to line up on what side of the center plane inside the cell. That's one way we can get genetic diversity and genetic recombination. But the other way that we talked about was something called crossing over. We talked about this. Crossing over was something that occurs during end of prophase 1 and right at metaphase 1 when the tetrads line up. And I mentioned to you that we have two sets of duplicated chromosomes that basically have 4 chromatids at play. So imagine this is my centromere. So just imagine there's another X down here. Here's my centromere here. So I've got a pair of sister chromatids here that represent 1 chromosome that's been duplicated. A pair of sister chromatids here that represent 1 duplicated chromosome. We know that when they come together in a tetrad, here's what's going to happen. There's going to be crossing over. The non-sister chromatids can actually wrap around each other and can exchange genetic information. What he saw in this ratio was basically showing the frequency at which these genes were doing that. So in the case of these genes, the genes between the eye color and the length of the wings for the fruit flies, because of these ratios he was getting, he was essentially discovering that 90% of the, as I mentioned, 90% of the offspring looked like one of the two parents, but 10% didn't. The 10% value represents the crossover frequency. How often were those genes swapping information between essentially in the original parent who was creating the gamete? How much of the information was being swapped from their mother and their father's genetic material? So in this case, he had 9 plus 1 plus 9 plus 1 equals a total of 20. 18 out of 20 looked like the original parents. They were the parental types. And then he had the 2 out of 20, the 10%. They were called the recombinant types. And the recombinant types, the amount of them in the offspring population is essentially indicating what the crossover frequency is between those two genes. So in this case, the reason why the ratio wasn't the same as he was expecting, the 1-to-1 to 1-to-1 ratio, is because the genes in question were on the same chromosome and they were being crossed over during prophase 1 or metaphase 1 during meiosis 1. That's the reason why the ratios weren't coming out. So this was something that Mendel had not come across, had not discovered. So because all the traits that he had chosen, they were on separate chromosomes. So there was no possibility of those traits in the pea plants crossing over and screwing up his ratio numbers. In the case of Morgan's research, he all of a sudden discovered that certain genes on the same chromosome would potentially cross over, which would change the ratios in question. So that's sort of what he came up with. So again, what I mentioned here, and this is sort of the recap here, you've got essentially, you've got 45% that were looking like the one parent, 45% that were looking like the other parent, and then 5% and 5% that did not look like either of the parents. That total of 10% was the recombinant types in the offspring that would lead a scientist to understand that there was a 10% crossover frequency between those two genes on the chromosome. Let me run this another way for you. How would you figure it out if you were just given numbers? Now, I'm going to erase part of the board here, just so I've got some room here to work with. So let's say, for example, I'm going to cross over. I'm going to do a breeding of these organisms. Okay, we're going to pretend like my results are going to be different than Morgan's because I want you to do some calculations. So we're just going to use sort of the same example, but be aware that that example is not going to be accurate to the actual results that Morgan got. But what I'm going to give you is I'm going to give you a bunch of numbers in the offspring. So here's what I've got. So out of this crossing, what we discover here is we're going to find out that in our offspring, purple vestigial offspring are going to be totaling, let's say, 356. And then I've got normal offspring that are going to total, say, 352. I've got some purple normal offspring that are going to represent, we'll say 148. And then I'm going to say there's some normal vestigial offspring that are going to be, let's say, roughly 144. I'll try to make the math a little bit easy for us here today. So these are the results of when I crossed these two parents. So I had a parent that was purple vestigial and a parent that was normal. I bred them together. I'm expecting to see a ratio of 1 to 1 to 1 to 1, or 1 to 1 to 1 to 1. Yeah, I said there were 4 of them. But I don't get that. I'm not getting 25% of my population. You can see that based on the numbers. I'm not getting that percentage. What I do have, though, is a total. I got a total here. So what I'd like you to try to do right now, based on these numbers, is can you calculate the crossover frequency of the genes that I'm dealing with based on these offspring numbers? How would you actually try to calculate that right now? Think about it. Pause the video if you need to. See if you can figure this one out. I'm about to give you the results right now, and then I'm going to show you how to do it. All right. Well, here's how you do it. It's on the next slide. If you were at home and paying attention, you can kind of calculate this fairly easily. What you want to do is you want to basically calculate the number of recombinant types in the offspring divided by the total number of offspring, because that's going to give you a percentage of the recombinant types in the entire population of the offspring. So I got to first of all figure out what my recombinants are. My recombinants are these ones here that don't look like either of the two parents. They don't look like the purple vestigials. They don't look like the normals. They got combinations of both. So I'm going to basically have 148 plus 144, and then I'm going to divide that by 148 plus 144 plus 356 plus 352, because that's the total amount of all my offspring, right? That's all of the offspring in play. So what I'm going to do now is I'm going to run my calculator here. I got 148 plus 144 is 158, so that's my total number of recombinant types in the offspring. I got to make sure I remember that. Let me do this again. I don't think I did that right. 292. That's the number of my recombinants in the offspring. It didn't sound right. So that's my total number of recombinants. 292, which basically gives me a recombinancy frequency in percent of 29.2%. So that's how you calculate the recombinancy frequency if you've been given the offspring numbers. You have to basically calculate, you have to determine which of the offspring are the recombinants, and then they become the top of your percentage, and then you have to divide that by the total number of offspring. That would include all the recombinants plus all the parental types. You just basically take that number, divide it by the total, and you're good to go. And that formula basically right there on that slide. So that's how you calculate recombinancy frequency. Now, what can we use? What other than the fact that it's telling us that our ratios are not correct, or they're not the ratios that we're expecting? What can we use recombinancy frequencies for? Well, there is something that we can use. What Morgan discovered in his experiments was this. He recognized that the possibility of two genes crossing over actually increased the further those two genes were apart from each other on the same chromosome, which kind of makes sense, because if I've got here's my tetrad at meiosis 1 about to go to anaphase 1, when they cross over, right, the non-sister chromatids cross over, it's much more likely that a gene up here and a gene all the way down here are going to cross over, as opposed to if I have a gene right here and a gene right there. In order for those two genes to cross over, the actual crossing point has to happen directly between those two genes, very close together on the chromosome. So he was able to determine that there was an actual correlation between the crossing over frequency of the genes on a single chromosome and their distance apart from one another. And what he devised it as was a unit known as a map unit. So what he found out was this: the distance between the genes on a single chromosome is equal to their crossover frequency. So therefore, what he called the map units, and that's just a generic unit of measurement on a chromosome. He measured it, by the way, he created his own unit of measurement called the centimorgan. So these map units are measured in an actual unit known as centimorgans, because again, chromosomes are very, very small, but we just refer to them mainly as map units. But the map unit number refers to the crossover frequency. It equals the crossover frequency. So for example, in the example that I gave you at the beginning of the lesson today, when Morgan did his crossover frequencies and he got that 45, 45, 5, and 5%, he had a crossover frequency of 10. So that means that in that example, the distance between those genes on the chromosome would actually be 10 map units. And because if you could calculate the crossover frequencies between two different genes on a chromosome, you could begin to create what's called a chromosome map. You can measure and you can draw and indicate the relative position of genes on a chromosome by how frequently they are crossing over during particular matings, during dihybrid crosses. So, for example, at the bottom here, now I don't know if you've got your notes with you, but if you have your notes up right now, there's a diagram there, and that's a chromosome map of a chromosome, and it's got all those different traits on it that are basically representing things. So the question beside it says, in the example to the right, what would the expected recombinant frequency of the genes controlling the limb length and body color be? So you've got to indicate, you have to find out where the genes are that can do this. So I can see that my body color gene is going to be at position 48.5, and then I can see that my limb length is going to be controlled at a gene on, I got to find him here, at 31.0. So if I've got 48.5 subtracted by 31.0, I should have the map unit distance between those two genes is going to be, if my math is correct, it's going to be 17.5 map units. That would correlate to, I would expect to see between those two genes, those two traits, I should see a crossover frequency of roughly 17.5%. So that's how you kind of use it. Now, to kind of expand beyond that, you can basically draw maps of the chromosomes, hence the term gene mapping or chromosome mapping. So, for example, on the slide here, I don't have it up on the board, but it's on the slide here. I've got, I'm going to give you the crossover frequencies of 3 genes. So if you haven't got your notes up, maybe grab them right now. If you can't read what's on the screen there, maybe enlarge that on your screen. but I'm giving you the crossover frequency. There are 3 genes that are located on a single chromosome. So I'm going to draw the chromosome right now. Now, if you've got the rest of the page open, you can already see the answer. So hopefully you're not cheating, but if you are, I guess you'll see my thought process between it. But here it is. I got a crossover frequency between genes A and B of 22. I got a crossover frequency between genes A and C of 10. I got a crossover frequency between genes B and C of 12. Can you draw a gene map or a chromosome map to indicate the relative position of these 3 genes? So you can pause me right now if you want. See if you can calculate. And again, the answer is on the page. The good news is on the next board here that I'm going to put, I'm going to give you an example that isn't in the notes. So you are going to have to actually run some calculations to see how I do it or how you do it. But this is what I've got right now. Pause me if you need to. Calculate what you need to do. And here comes the answer. What I'm looking for here is I'm looking for patterns in the math. I can see that my largest number is 22. So to me, A and B are going to be the furthest genes apart from one another in this chromosome because they have the highest crossover frequency. So if I put A over here and B over there, that's fine. Could I put B and A? It doesn't really matter. The key about this is it's a relative position. So could I draw the gene map backward or forward? And is it still right? Absolutely. It's about a relative position rather than is it right from left to right or right to left. It's just what's the relative position of all the genes on the chromosome. Now, here's a pattern that I'm seeing. I see that A to C is 10 and B to C is 12. Well, 10 plus 12 is 22. This is a pretty easy one because there's only 3 genes. So I know that C is going to be somewhere between A and B, but I know that C is going to be slightly closer to A than it is to B because if I have C here, C would be there, which means that this distance now is 10 map units. That would be 10 map units there. C to B would be 12 map units over there. And then my B to A is going to be the addition of both of them, 10 to 12. Be aware the numbers don't always match up perfectly. You might have numbers that are off by like 0.3, 0.4, 0.5, but you're looking for general patterns in the math to see, do I have numbers that add up to other numbers in the list? Obviously, the more genes you have on a chromosome, the tougher the question is going to be. But that's the kind of questions you would get on a diploma, on a quiz, on a unit exam. These are questions that we expect you to know how to do them, how to either draw them or interpret the data based on these crossover frequencies. And there you go, up on the slide, there's the actual chromosome. Now I'm going to give you one that I'm going to pause it right there. I'm actually going to pause it right there. I'm going to go back. Actually, we don't need that slide because I'm actually past my point of no return. Let me go back. Yeah, we'll go back to there. There we go. We'll go back to that slide. The next slide, that's for the next day. I'm going to finish off with no more slides. This is going to be an example for you. Here's an example that I want you to try. It's not in the notes. It's only up on my whiteboard. If you're paying attention watching this video, you're getting some extra help here going into the booklets that are going to be assigned for today. I got 4 genes that are located on a single chromosome and have the following crossover frequencies with one another. Gene A to B, the crossover frequency is 10%. Gene A to C is 25. A to D are going to cross over at a 23% crossover frequency. B to C is 15. And C to D are going to cross over at a rate of 48%. What I would like you to do is take genes A, B, C, and D, and I want you to draw their relative positions to each other on a chromosome to draw a chromosome map or a gene map, okay? I'm going to draw the chromosome right now. There's my chromosome. And I'm going to have 4 little ticks there that are going to represent the 4 gene loci. We talked about position of genes are called the loci or the locus. There's a single position of a gene, a locus. Give me the 4 gene loci or loci on this actual chromosome. Pause me right now. I'm going to give you the answer in about 2 seconds. All right, here's how you calculate it. I'm looking for my big number first. D to C, 48%. They're going to be the ones that are going to be the furthest apart from one another. Let me start with that. So I'm going to go C and D. I'm going to put D out there, C over here. That's a good starting point. Now I'm looking for patterns with the numbers. Do they match up? Do I have 2 numbers here that match up to 48? Yeah, I do. 25 plus 23 equals 48. That means that C is going to be roughly halfway between, A is going to be roughly halfway between D and C. It's 25% to C, or 25 units to C, 23 units to D. So it's going to be slightly closer to D. So we're going to put A right over there. There's A. Now I got to figure out the position of B. Now I'm going to start canceling off the information that I've already used. That also makes my life kind of easy so I know which genes I've dealt with. These now are the last two bits of information. Do those numbers add up to any other number that I've got up on the board? Yeah, they do. 10 plus 25 equals, 10 plus 15, 10 plus 15 equals 25. And that makes sense because it tells me that gene B is going to be between genes A and C, and it's going to be slightly closer to A than it's going to be to C because it's only 10. It's got a crossover frequency of 10%, which is going to be 10 map units. So I'm going to put my position for B over there. So that's my relative gene map for the chromosome in question. Can I run all the math to make sure that all the math is correct? You certainly can. I got 15 map units to there. I got 10 map units to there. A to D in theory, or A to D is supposed to be 23. C to A is going to be 25. So that works out okay. And then my C to D is going to be equal to, let me just erase that, is going to be equal to 48. So all the math there works out, right? 15 plus 10 plus 23 equals 48. 25 plus 23 equals 48. All the math is correct. Relative positions are all good. That's how you do it. That's gene mapping in a nutshell. What's your homework for today? I got a couple of sets of questions for you today. I've got a gene mapping multiple choice question booklet that I'd like you to go through. They're old diploma exam questions that are related to gene mapping, so give that a shot first. I would do that booklet first. Once you get your teeth, you kind of sunk your teeth into those multiple choice questions, I got some short answer questions for you in another booklet just called Gene Mapping Question Booklet. So 2 sets of booklets here due for our next class. Until then, thanks for tuning in. And next day, I'm going to be discussing X-linked genes, how some genes are actually found on the X and the Y chromosomes, and how that can affect inheritance of those particular traits. And I'm also going to discuss something known as multiple and lethal alleles. Until then, thanks for tuning in, and we'll see you soon.